using fitness and environmental patterns to identify beneficial alleles in maize landraces. So when we're talking about local adaptation and identifying the alleles that underlie local adaptation, there's a few different patterns that are very characteristic that we can start to utilize. And one of them is if you take organisms from disparate environments and then you grow them in a variety of environments, and you see that organisms, when grown in their home environment, have higher fitness than non-native environments. That's a pretty characteristic pattern, as illustrated here by the Clausen-Keckenheise example, where you can see sort of this local home site advantage. But in terms of identifying what are the adaptive alleles or loci that are leading to this pattern, if you think about having just a whole genome worth of SNPs, what pattern would you look for? And it would probably be something similar to this simple quantitative model here, where you're looking for one allele that has a fitness benefit in one environment, and then an alternate allele with a fitness benefit in another environment. And so the second pattern that we can look for is adaptive alleles that show allele environment associations. And so this is from the Dobzhansky 1948 paper here, showing an environmental gradient going up the Sierra Nevadas, and these boxes represent allele frequencies, and you can see that the black box here at the highest frequency in the lowest elevation is actually at the lowest frequency in the highest elevation. And again, when we're thinking about, now I have all of this, say, genome-wide SNP data, what pattern am I looking for? This one's a pretty simple one. Over a gradient of environmental change, we should essentially see allele frequency tracking that. So now I work in maize landraces, and maize landraces, they're open pollinated, so very different from kind of the inbred hybrids that we typically grow or see growing here. And since they're open pollinated, they're allowed to adapt to their own environments, and they're grown over a tremendous diversity of environments, from the mountaintops to the temperate plains, all the way down to sea level, deserts, the tropics, you name it. And then illustrated by this figure here, they have a tremendous amount of diversity that adaptation can work with. And so if we take just a handful, naively, of really diverse maize landraces, and then we start to grow them out, one of the most characteristic things that you're going to see is that temperate or high elevation landraces, basically any landraces that are adapted to a place that has an early winter, a short growing season, they tend to flower, they get their lifespan done like really fast. Meanwhile, tropical maize, they just kind of take their time. They're in no hurry. There's not a big point in that. And that really suggests that we can find these environmental gradients that we can then leverage to start to find allele frequencies that actually indicate where our adaptive loci are. And then another great thing about working with maize is everyone knows how to grow maize. We grow maize everywhere. And maize seeds are really easy to transport. So we can take seeds from highland plants and grow them in the lowlands, like this figure here. And again, we see this very adaptive signature where lowland plants are totally happy in the lowlands, highland plants over here, they're totally happy when we're growing in the highlands. So it's really a matter of taking like our big genome data and then starting to look for these patterns, and then we should be able to get a good idea of what the adaptive alleles are. And so this is the data essentially that I'm working with, or this is kind of the world that I live in. And the data specifically that I work with is from our collaborators at CIMMIT in Mexico, and it's just this tremendous effort that they've done. They've grown up hundreds of thousands of plants in this replicated F1 mating design. Once you correct for the replication and we start to do association analyses, we get down to having around 15,000 of these data points at 6 traits, and essentially what we get, the 15,000 of these data points represent one data point per individual accession across a number of different experimental trials. And so you can see here is the diversity of the individuals in black. That's where the accessions are from. So trying to cover the whole range, the environmental range in Mexico. And then the red is the trials that we've grown in, again, trying to really get a hold of the whole environmental range. And then, of course, we have our genome-wide SNP data as well. So just broadly in this data set, do we see signatures of adaptation? And this is at the whole plant level. This is altitudinal distance between plants' natal environment and a trial in terms of elevation in meters, because that's very important. And so 0 is basically like when you're grown in your home environment. And so you see that they take longer to flower, they produce less seed, and they're shorter. So just generally lower fit plants when you grow things outside of their home environment. And so we proceed to do our GWAS, our association analysis, again, sort of looking for this pattern, but in a much bigger, gnarlier, mixed linear model framework. And so this is, again, in these big data sets, when you run big GWAS on these big data sets, you start to get lots of these peaks, these outliers. But we wanted more than just to say, oh, well, we found peaks. We actually wanted to validate these. So what we did is we took 1,000 of these outliers here, and then we tested to see, does the allele that is predicted to do well in the highlands, does that actually live in the highlands? And we found pretty resoundingly yes, that the effects from this model predict where these alleles are found on the landscape. And even more interesting than that, we found that these effects actually predict allele frequencies in closely related teosynthase subspecies. We have this really nice example because the subspecies Mexicana prefers the highland, the subspecies Parviglumis prefers the lowlands. So we can say, do our maize landrace highland favoring alleles, do they exist more often in the Mexicana? And yes, they do. And this middle here, this is paired allele frequency, just randomly sampled alleles. So you can see this is kind of what we would expect if we had no signal at all. But again, lowland favoring alleles tend to be found in the lowland favoring subspecies, which is really incredible kind of in my mind, because this is 50 to 100,000 years divergence between some of these subspecies. And so it indicates that this is either standing genetic variation, because there are a lot of large population sizes, or possibly adaptive migration, because these often come into contact with the maize landraces. So the second pattern that we're looking for here is this allele frequency associations. Again, when we run our mixed linear models over looking for allele frequencies with environmental gradients, we get a bunch of peaks, and again, we can sort of go back and validate them and say, do these peaks, do the alleles that live in the highlands, do we expect, or do they increase, say, your yield when they're grown in their favorable environments? And so what this is essentially, if we grow alleles in their favorable environment, they have a much higher yield than just basically randomly sampled alleles. And this is for yield here. We ran into something interesting, though, is that flowering time doesn't really show this pattern. And so when we're going back and we're looking at this, we're essentially looking at the G by E effects. So do the G by E effects from this model essentially line up with the environmental GWAS that we have run? But we started to think that maybe flowering time is actually kind of an unusual trait. So in yield, you always want high yield, no matter where you are. Like if there's just something that always makes you low yield, it's not going to be around for long. But flowering time is really only important, fast flowering time in the highlands. So we actually went back into our models. Instead of looking at the G by E, we looked at the main effect, which is just the site independent or environmental independent genetic effects. And what we see is that fast flowering time can actually be achieved not really requiring G by E, because it doesn't matter what's going on in the lowlands. The only thing that really matters is that highland plants flower faster. And it actually doesn't seem to be as much that highland alleles are like really fast or have lots of really fast alleles. It tends to be that there are lots of slow alleles in the lowlands, and they just don't ever make it into the highlands. They're just selected against, they just stay down in the lowlands. And so that was a really neat result for us there. But we can extend this further because it's maize, and we should be able to extend this into now experimental trials. We grow maize in a lot of drought trials, that nature, and we see the same thing. Instead of looking at altitude now, we can actually change and look at precipitation and ask, do low precipitation alleles increase your yield in drought trials? And we do see this pattern again, and this is a really interesting place to be because I think there's a tremendous potential. Once you can start to get into experiments, you can really start to disentangle a lot of factors that you can't just kind of in multi-environmental trials. You can really start to hold a lot of things constant. And so this is an exciting place for us. And the one other nice thing about, like, when you start to get in experimental trials, we can really start to nail down really strong candidate alleles, because one of the things that we're interested, especially when we're talking about drought, is moving some of this adaptive variation that exists in land races then over into breeding populations to possibly help make more drought tolerant, more climate ready production maize. And so this is an example. I'm going to walk you through this gene glossy 15 here. And this is the low precipitation allele is actually, so from our environmental TWAS that we have run, is actually increases the yield pretty dramatically based on these estimates here in our drought trials. So that's a very good validation that it's a pretty strong candidate. But again, it's maize and it's drought trials, so there's tons of RNA sequence data floating around out there, and we can go even further and say, do we also see differential expression of this candidate that would make it a stronger candidate? And we do see differential expression here in the reproductive tissue. The expression is severely reduced in drought. And then we can even get so many of these candidates that we can actually find ones that we have known function for. Again, like this one, glossy 15, is kind of this classic mutant, and one of the reason that it's called glossy 15 is because variation messes with the epicuticular wax, basically the wax outside of developing seeds, and that is tremendously important when we're talking about water use. We're talking about developing maize in drought. Now we have different variation in these waxes. And so now we have a clear functional link going forward. We're thinking about, obviously, CRISPRing and doing all sorts of crazy stuff now that we're starting to get these candidates. But I think this really illustrates a diversity of the different things that you can find out when you start to really nail down these adaptive alleles. So we find that the increased fitness is found in their favored environment, even across subspecies. That flowering time adaptation consists heavily of depletion of slow flowering alleles in the highlands, and that also experimental manipulation drought trials, that they offer a really good way to further refine our candidate. And I think that there's a huge range of possibilities now for us to start to get in and really nail down a lot of this stuff here. And so with that, thank you my collaborators, number of institutions, and then the Rossi Bar Lab. So thank you.